Okay, in this video we're going to talk about the reaction of glycolysis, and then the steps, and then some of the more important, the regulated steps, as well as substrate level phosphorylation. Right. So in a lot of this, I'm gonna abbreviate since my workspace is really small. So the idea is that we start with glucose, and we convert it to glucose 6-phosphate. Right. Now the enzyme here that most of you guys know is hexokinase. Right? I know I spelled that wrong, but you get the idea. So hexokinase, and then there's actually another enzyme called glucokinase, which is similar to hexokinase. All right, and this enzyme is more prevalent when blood glucose levels are high. Right, now properties of glucokinase is that it has a high Km and it also has a high Vmax. Hexokinase, on the other hand, has a low Km. Right, so again, irreversible step to glucose 6-phosphate. Glucose 6-phosphate, through an isomerase reaction, get converted to fructose 6-phosphate. Right, the enzyme in this step is actually phosphohexose isomerase. Right, now this is actually a reversible reaction. In the next step, fructose 6-phosphate gets converted in the most important step in glycolysis to fructose 1,6-bis phosphate. I'm not going to write here all of it out. Right, now the enzyme that does this is a long one, and it's fruktokinase 1, or in other words, it's called PFK1. And so phosphofructokinase, or PFK1. Now this step is the most important step of glycolysis because it's inhibited or upregulated by a couple of things. Right, so phosphofructokinase is actually downregulated by the presence of increased ATP as well as citrate. In the same breath, it's upregulated by AMP and fructose 2,6-phosphate. Right, so this is why, and we're going to talk about the reasons in a different video, but this is why this is the most important step of glycolysis. Again, this is an irreversible step. In the next reaction, we convert fructose 6-phosphate using aldolase A or B. There's actually two aldolase, A or B, and that's converted into glyceraldehyde 3-phosphate, which I'm going to abbreviate as G3P, and dihydroxyacetone phosphate, so DHAP. Now, allulose A is important for the muscle. So allulose A is actually in muscle, whereas allulose B is used for the metabolism of fructose. Right? So that's the difference. Again, for maybe an undergrad institution, you guys just probably need to know that there's just allulase, right, that cleaves them. Now, glyceraldehyde, right? So at this point, we have a 6-carbon sugar converted into 2 3-carbon sugars. Right? Now, what's important is that dihydroxyacetone phosphate has to be converted back to G3P in order to continue the glycolytic cycle. Now, as a side chain, dihydroxyacetone phosphate is actually used in several instances to form glycerol 3-phosphate. And glycerol 3-phosphate eventually leads to triacyl synthesis. right? So as a side note, dihydroxyacetone phosphate could eventually go to glycerol 3-phosphate that eventually leads to triacylglycerol synthesis, right? But most of the dihydroxyacetone phosphate gets converted back to G3P in order to continue the cycle. So now as a result, we have two G3P molecules, and so everything in the cycle now becomes multiplied by 2. Right, so 2 G3P gets converted, right? So 2 glyceraldehyde 3-phosphate dehydrogenase, 2 G3P gets converted to 1,3 bisphosphoglycerate. Right? Now, what's the enzyme? The enzyme for this is glyceraldehyde 3-phosphate dehydrogenase. So glyceraldehyde 3-phosphate dehydrogenase. Now, you could remember now, I missed a step here, right? I missed both steps. I'm going to put it in later. But in glucokinase, in glucose to glucose 6-phosphate, I actually use ATP in this step, right? So this is my energy investment phase, as well as fructose 6 as well as fructose 6-phosphate to fructose 1,6-bisphosphate. I'm going to use ATP as well. So my end product is ATP, right? And again, those two are the energy investment phase. Going back to 1,3-bisphosphoglycerate, using glyceraldehyde 3-phosphate dehydrogenase, in this step we actually create NADH plus H plus, and we use NAD as a cofactor. Now this step is actually reversible. From 1,3-bisphosphoglycerate, and I'm going to circle this. This is a high energy compound. We're going to get substrate level phosphorylation from this, right? In a reversible reaction, right? So from 1,3-bisphosphoglycerate, we're going to create ATP, right? And so it is one of the high energy yielding compounds in glycolysis, right? So from this, we create ATP by substrate level phosphorylation, and the enzyme is phosphoglycerate kinase. Right? So the enzyme here is important, as well as the high energy compound. From 1,3-bisphosphoglycerate, we get 3-phosphoglycerate. So I'm just going to write 3-phosphoglycerate. Right? So normally we get 3-phosphoglycerate, right? And then from 3-phosphoglycerate, we get 2-phosphoglycerate. Again, this is a reversible reaction. This is 2-phosphoglycerate. And the enzyme that does this reaction is phosphoglycerate mutase, right? So this is done by phosphoglycerate mutase, right? From 2-phosphoglycerate, we lead to phosphoenol pyruvate. Phosphoenol pyruvate. I'm going to circle this as well. This is the second high energy yielding compound. This is done by enolase. Now fluoride actually inhibits enolase. And the idea is that whenever you go to the doctor and you get your blood drawn, the capsule that your blood is placed in actually contains fluoride. And the reason being is that they're preserving the glucose, so the the glucose levels doesn't become abnormally low, right? Because as a result, if you do not add fluoride, what happens is that the blood that they have drawn spontaneously goes through glycolysis, and again, as a result, they can't measure whatever stuff they want to measure, right? So if they were trying to measure your blood glucose levels, right, it wouldn't be appropriate to have all the glucose being utilized by glycolysis, right? So fluoride is one of the elements that inhibits ENLAs and preventing glycolysis from occurring. Similarly, arsenic, right, so arsenic poisoning actually inhibits, and I know it's kind of messy here, but watch me, arsenic poisoning, right, so arsenic poisoning actually inhibits glyceraldehyde 3-phosphate, right? So we talk about arsenic poisoning inhibits glyceraldehyde 3-phosphate. Now, from phosphoenol pyruvate, we lead to the last step, and again, this is a reversible reaction. So from phosphoenol pyruvate, we lead to 2-pyruvate. Remember, 2 molecules of pyruvate, and this is done by pyruvate kinase. Now remember, this is 2 molecules of phosphoenol pyruvate. I have 2 molecules of phosphoglycerate kinase. I have 2 molecules of 3-phosphoglycerate. And I have 2 molecules of 1,3-bis phosphoglycerate. Remember, everything gets multiplied by 2. So 2G3P, 2 1,3-bis phosphoglycerate, 2,3-phosphoglycerate, 2 phosphoglycerate molecules that eventually leads to 2-phosphoenol pyruvate, and eventually two molecules of pyruvate. Right? So let's have a little discussion real quick. The substrate level, there are 2 substrate level phosphorylation steps in the glycolytic pathway, and I've circled the high energy compounds, or 1,3-bis phosphoglycerate, so let's put a star there, and phosphoenol pyruvate. So 1,3-bis phosphoglycerate and phosphoenol pyruvate. Now, with substrate level phosphorylation, only one of the reactions is actually irreversible, and that's the reaction that consists of phosphoenol pyruvate going to pyruvate. The other substrate level phosphorylation, again creating ATP, which I did not put here, but again ATP from ADP is 1,3-bis phosphoglycerate to 3-phosphoglycerate, again creating ATP. However, this is a reversible reaction, right? So this is irreversible while this is reversible. Again, they're both substrate level phosphorylation. Now, what does that mean? That is important because that means that glycolysis can still occur even in the absence of oxygen. So that's why substrate phosphorylation is important, right? Now let's talk about the regulated steps in glycolysis, or the regulated enzyme. What do we mean by the regulated enzymes? The enzymes that catalyze reactions that are irreversible. So glucokinase, hexokinase. So we talk about we already talked about the substrate level phosphorylation, but the regulated enzymes, right, should be your glucokinase, hexokinase. So that's one. The second regulated enzyme is phosphofructokinase 1, right? So we have 1 here and we have 2 here. Again, it's an irreversible reaction, right? So the next regulated enzyme would be phosphofructokinase 1. The third and the last one would be pyruvate kinase, right? Because it's catalyzing an irreversible reaction. So this would be pyruvate kinase. All right. And ladies and gentlemen, we've just went through the process of glycolysis.